When we think of plastics in the ocean, we often think of a plastic bag or plastic bottle floating. Though that sometimes may be the case, these plastics are broken down into smaller and smaller pieces known as microplastics. Microplastics are heavily prevalent in our world today, and they've even been found in a variety of places, from fish brains to human stool samples. My project this year is a continuation of previous science projects involving microplastics. My first project looked at the presence of microplastics in ocean water samples from the southern Maine area, with the results showing high numbers. My second project looked at the impact of microplastics on mammalian cell culture, with an overall negative impact being determined, given a decrease in cell growth and an upregulation in stress and inflammatory markers. Converting my living room into a lab, I developed a test using glass syringes, Nile Red, a membrane filtration device, nitrocellulose membranes, an orange camera filter, and a crime light 2, which is a handheld device that emits different colored lights. The one that I used was specifically a blue-green light. Using a positive control with known concentrations of polystyrene microspheres and a negative control of pure water, my test accurately detected the presence of microplastics with a sensitivity of above 100 microgram per mil. I then used my test on real-world water samples from 4 beaches and 5 different brands of plastic bottled water, with microplastics being detected in 2 of the ocean water samples and 1, surprisingly the most expensive brand of bottled water. Overall, I was able to find a method to rapidly test for microplastics within 20 minutes. Further research would include making a more rapid, at-home test with easily visible results, which could be done with gold labeling and conjugated Nile Red.